Good morning, good afternoon, and good evening. Thank you for being here, and thank you for giving us this opportunity to share with you and bring another workshop here for you. And this workshop is on model-informed drug development, and we have an expert scientist here with us today, Dr. Rick Lalonde, who is currently at the University of Florida. And prior to this, he actually worked for many years at Pfizer. And it's our honor and privilege to host him this morning. So to tell you a little bit more about us, Project Dontabhaktuni. Project Dontabhaktuni is hosting today's workshop, and today's workshop is sponsored by PharmaPro Consulting. Let me introduce myself. I am Aruna Dontabhaktuni, the founder and CEO of PharmaPro Consulting, and the founder and scientific director of Project Dontabhaktuni. So I have over 26 years of experience in drug development. And in my career, I have worked in India, in the UK, and now in the US, and have supported over 60 plus compounds through various stages of drug development. So to tell you a little bit more about Project Dontabhaktuni, this is a nonprofit organization created to support scientists and students, postdocs from around the world. And by creating such workshops, training programs, and mentoring programs. So how can you support us? You can support us by joining hands with us, by participating in these workshops, and by being part of the mentoring programs, either as a mentor or as a mentee. And thus our motto, which is empowering you to empower others. And our request is that you actually take whatever you're learning from here and teach at least one more person. So knowledge grows only when you share. So what have we done so far? This is our second year running. So in 2019, we actually, so let me actually talk about 2020 first, and then I will get to 2019. So in 2020, while the world was dealing with the pandemic, we actually took on doing a lot of workshops similar to what you're seeing here. And we had 3 workshops, and majority of these workshops had about 500 or so participants from around the world. Some of you may have attended those workshops. Thank you for being there, and thank you for continuing to be part of our outreach. And in 2019, a few of us had visited India and China and hosted on-site workshops in many universities. And we are really proud to share that in the past 2 years, we have reached over 1,600 students and scientists from around the world. So to continue the same efforts, this year we had hosted a prior workshop, and we will continue to do the same for the rest of the year as well. In fact, at the end of today's workshop, I will tell you a little bit more about a 3-month certified pharmacometrics program that we would be starting from June through August, in which we will cover some fundamentals of pharmacometrics, also teach some fundamentals of R, and share with you about an upcoming biosimulation tool called RSNLME from Certara. So without further ado, I Also, yeah, before we actually move on to talk about today's talk, wanted to give you an idea about who are the various participants who are here with us. So we have about 142 participants, and majority of them have PhDs. And this is just an overview of all the information that you have provided to us while you registered into this workshop. And also really excited to share that we have participants from 26 countries in today's workshop. And so the 44% that you see here are from India, 31% from United States, and then we have multiple other countries represented. So 26 countries here. Wow, I'm really amazed. So without further ado, I would like Dr. Partha Nandi to introduce our speaker today, Dr. Rick Lalonde.
So thank you, Aruna, and a big thank you to Richard for accepting the invitation to speak to the group here today. So in terms of introducing Richard, who goes by Rick, I've known him as Rick throughout my life here in the US and in career as a clinical pharmacologist. He is currently an adjunct professor at the Center of Pharmacometrics and Systems Pharmacology, which is under the Department of Pharmaceutics, College of Pharmacy, University of Florida. And he is currently working as a consultant to several pharmaceutical industries. Previously, he was with Pfizer for 19 years, where he served as the vice president and global head of clinical pharmacology for the last 11 years of his tenure until he retired in 2017. Prior to joining Pfizer, Rick was the scientific director and head of clinical pharmacology at Phoenix International in Montreal between 1991 and 1998. From 1994 to 1991, he was assistant professor and then a tenured associate professor and vice chair for research in the Department of Clinical Pharmacy within the College of Pharmacy, University of Tennessee at Memphis. Richard is a fellow of American College of Clinical Pharmacology, American Association of Pharmaceutical Scientists, and American College of Clinical Pharmacology. He was president of the American Society for Clinical Pharmacology and Therapy, in short ASCPT, in 2011 and 2012 year, and member of its board of directors from 2010 to 2013. In 2021, Richard was recipient of the Oscar Hunter Award in Therapeutics from ASCPT. He also went on to receive the Gary Neal Prize for Innovation in Drug Development from ASCPT in 2017 and International Society of Pharmacometrics Leadership Award in 2014. He has been a member of editorial boards of clinical pharmacology and therapeutics since 2005, prior to serving as associate, subsequently serving as an associate editor 2007 to 2010. He served on the National Advisory General Medicine Sciences Council at the National Institutes of Health, Bethesda, Maryland from 2013 to 16. Richard has been invited speaker at various national and international meetings of professional societies, universities, government, regulatory agencies in the US, namely FDA and NIH, in Europe, the Medicines Agency, in Japan, PMDA, and in China, CFDA. Over the past 40 years, his work has been focusing on the quantitative application of pharmacokinetics and pharmacodynamic principles to the optimal development and utilization of drugs in patients. Richard is a graduate of University of Minnesota, from where he received his PharmD degree, and he also holds BSc in pharmacy from University of Toronto. Today, it's with great pleasure and honor I invite Rick to present to this group, which summarizes all the knowledge and work that he has been involved with and led many of this work over the past many decades. And his talk today is the model-informed drug development and how to utilize this technique in furthering and expediting the drug development process in industry and academia. So thank you, Richard, and I'll hand it over to you.
Thank you very much, Partha, for those kind words. Thank you for the invitation, and good morning, good afternoon, and good evening to everybody around the world. It's a privilege to participate in this webinar. So we'll be talking about model-informed drug development. And very briefly, this is a quick overview. We'll talk about drug development risks, some of the common biases in decision-making under uncertainty that we all have, and we'll discuss drug development as a model building process and the elements of model-informed drug development, or MIDD. And I'll spend most of the time discussing some of the examples of the application of MIDD. I believe we'll have time also at the end to answer some questions that you may have. I think I'm also told that there are some clarifying questions during the webinar to make it clear. If you need a clarifying, if you have a clarifying question, we can entertain them during the presentation. So whatever works better for everyone. This will be focused mainly on examples, not as much on the technical details, but really on the impact of model-informed drug development. So, as you probably all know, drug development is a risky business. About 90% of compounds fail from first in human to regulatory approval. And while those were older data, these newer data essentially show a similar trend. Circled in right here is the likelihood of approval from phase 1 all the way to registration, and focusing on the blue bar, about 10% make it through. So again, about 90% failure. We won't go into the details of why that is. It's a complicated story, but the point is that it's a very risky process. Drug development is very lengthy, and most of the compounds fail. So we have a lot of risks involved. And I often show this slide here to illustrate how this is similar to a race. Let's say it's a 400-meter race around the track, and for every winner, there would be many losers. And the odds are that the compounds you're working on in drug development will not make it through to the end of the race, or will not win the race. So this is really a challenge, if you wish. Going back to the race analogy, will be how do we select a potential winner early on in the race where there's a lot of uncertainty. And this is about a field that is actually a very interesting one, about the flaws in decision-making with uncertainty. As it turns out, there's a lot of famous academic research done in this area. Pictured here are 2 Nobel laureates, Daniel Kahneman from Princeton University in 2002, and Richard Thaler from the University of Chicago in 2017, who received the Nobel Prize in economics for their work really on the discipline that we now call behavioral economics. And the key part of their work is really how we all have common flaws and biases in how we make decisions. And this impacts all kinds of decisions, whether it's about how we choose investments, for example, and the biases, and that's why they were recognized in the field of economics. But this is how you may make decisions on how you get your news, how you pay attention to certain things more than others, and also how we make drug development decision-making. This paper, which is very well known in this discipline, that's from Science, long time ago, but summarizes some of those key biases and heuristics. Heuristic is simply a mental shortcut that we use to essentially make a quick judgment on things. So some of those biases that are described here is one is anchoring, which is the tendency to rely too heavily on the first piece of information that's offered, the anchor. We see this all the time when people are, for example, bartering on the price of something. You may offer a very low price. Someone asks for a very high price to try to get you to move towards their particular offer. But we see this all the time in how people plan things. I don't have time to go into detail, but this is a common reason why projects often take longer and cost more money than originally anticipated. The availability bias is a mental shortcut that occurs when people make judgments based about the probability of events by the ease with which these examples come to mind. And a good example of this is when we're watching the news and there's something that's very newsworthy, like maybe the crash of an airplane or an earthquake or some whatever other unfortunate event. We tend to overestimate the probability of these events affecting us because they are very available to us based on this recent news, for example. Overconfidence is also very common. We tend to overestimate our ability to affect future outcomes, and we take credit for past outcomes and neglect the role of chance. This happens a lot in drug development, and I'll come back to this in a minute. Confirmation bias is maybe the most important one of all. This is where we make selective use of data that fit our preconceived ideas. And in clinical drug development, for example, this may be a situation where we accept or put more weight on a positive trial A that may fit our preconceived ideas, but we'll find ways to dismiss the negative trial B because of whatever reason we can come up with. So trying to find information that confirms our point of view. We see this all the time in how people consume their news these days also. The representativeness is the insensitivity to sample size. For example, if we have a proof of concept study with a relatively small sample size or a larger sample size, we tend to neglect the uncertainty, or we ignore the uncertainty in the estimate of, let's say, the mean response. And this is probably a common reason why we overestimate our the likelihood we can replicate study results. If we have one study that we completed today, we do another similar study next year, we overestimate the likelihood that the results will be essentially the same as the original study. In another publication by Kahneman with another collaborator in the Harvard Business Review, they further talk about the delusions of success and how optimism undermines decision-making. This is a very common flaw again. As I just mentioned a minute ago, we tend to overestimate our ability to make good decisions with uncertainty. We take credit for positive outcomes. Negative outcomes tend to be blamed on external factors. So we say, well, we made all the right decisions when things go well, but if something happened that a project didn't go forward, for example, we say, well, there was some external factor. It's not because we made a wrong decision, but something happened to derail the project. So we tend to ignore these things, or we dismiss them, but we overestimate our ability to make good judgments. There's some interesting books on this topic, Fooled by Randomness by Nassim Taleb, and Thinking Fast and Slow by Daniel Kahneman are interesting books if you want to read more about this. In a completely different discipline, there's a book called Moneyball by Michael Lewis that was published almost 20 years ago now, that talked about the quantitative use of prior data in baseball. This essentially, this analysis was based on how this debunked the so-called expert judgment that had been used for decades in assessing the value of a professional baseball player based on statistics. And it turned out that these statistics were really not the best guide of the value of a player. So basically a better use of the prior data was useful here. So there's actually was turned into a movie that came out 10 plus years ago. So in drug development, we have teams that may work on a project for many years. So coming back to this optimism, we expect these teams to be optimistic, and this goes with the territory. It's actually good because it provides motivation, creativity. We also know that there will be some inherent biases in organizational pressure to accentuate the positive. So on a team, for example, when you're trying to get a governance group to support your project, you will accentuate the positive because you may be competing for finite resources with another team that will be presenting in the next hour. So you want to show the best possible side of your project. So this leads commonly to overestimate the probability of success of compounds. This is very evident. We've seen this all the time. In my experience in drug development, we see this in big companies, small companies, especially notable in small companies where the success of a company could be based on the value of one particular compound. This is to contrast. Now, I've talked mainly about drug development teams where we have this optimism. A governance committee, on the other hand, should be looking at these principles of decision-making with uncertainty and how to mitigate these biases, because the job of the governance committee is to, at the portfolio level, look at how they should support project A and B, but not project C and D, for example. So they need to try to dissociate themselves. So over the years, what I've learned, what others have learned, is that we can't really expect drug development teams not to be optimistic and not have some of these biases. It's part of human nature. We all have them. To some degree, it is good. However, for decision-making by a governance committee, we need to have ways to mitigate some of these biases. As it turns out, in this paper, in Harvard Business Review, they talk about what they refer to as the outside view. But in drug development, essentially the way to mitigate these biases, or a way to mitigate these biases, is the quantitative use of prior information. And we have a lot of this in drug development. We have a lot of prior knowledge about our current compound, related compounds from the same class, competitors. And really what we want to do is look at the totality of this information and really get at the probability of achieving a given outcome based on all pertinent data. And this led us to, in 2007, to write a paper summarizing some of these principles in what at the time was called model-based drug development, but really talking about the continuum of drug development from the preclinical phase through registration and post-approval, that we're continuously learning throughout this process. And we use, in these different phases, modeling and simulation, M&S here, where we may be developing a model based on the existing information we have. We then simulate what we expect to see in the next phase of development. Then we update our model based on the new information that we've gathered. And there was new model, now we can simulate what we'll expect to see in the next phase of development, design the studies accordingly. And then we continuously go through this process, building over time. So thinking of drug development as a model building process, a continuous process over many years. And what we're trying to do essentially is manage the uncertainty, if you wish, but there's a lot of uncertainty at the beginning, and we want to have relatively much less uncertainty when we have to, for example, demonstrate to a regulatory agency that our drug is relatively safe and effective. And really, this is about building confidence in our drug and disease model. So we described these principles back in 2007. These were some of the components. As I said, at that time we called it model-based drug development, now more commonly referred to as model-informed drug development. Different types of components, if you wish. Others may have different ideas about these, and I'll spend a bit of time talking about some of these, the criteria, for example, the PK/PD models, the competitor and meta-analyses information over the next 40 minutes or so. So just very briefly, I realize we have a diverse audience here. Most of you are going to be very familiar with this, but just in case you're not, when we talk about the dose concentration effect relationship or the exposure response relationship, we're talking about related dose to drug concentration in the body. And this really is about pharmacokinetics and really concentration to the pharmacologic effect or therapeutic response. And really, this is the domain of pharmacodynamics. in a very simplistic way. What the body does to the drug, it would be pharmacokinetics, and what the drug does to the body would be pharmacodynamics. And we can have the interrelationship here. This is pharmacokinetics, concentration over time. This is pharmacodynamics, concentration versus effect. And linking the two, we can then relate drug administration to the effect over time, which is really what we're most interested in typically. So we can build then a model, which we call a drug and disease model, based on some of these pharmacokinetic principles, pharmacodynamic principles. But when we start out with a disease model, we'll incorporate maybe other bits of information, for example, about placebo effect, the effect of the disease, different covariates, to ultimately try to predict the response, incorporating variability in these parameters, uncertainty, et cetera, et cetera. So with these models, we can then look at, for example, the time course of response. In this case, in this particular publication, again, we looked at gabapentin in neuropathic pain. So this was a model-based analysis of the phase 3 clinical trials. So this is looking at pain score now over several weeks. So this is a typical phase 3 clinical, or actually different clinical trials, looking at the pain score. And what you can look at here is that we have a placebo effect that comes on over time. So this could be due to many different factors. Could be, for example, regression to the mean is probably a very important element of this. And we can look at the drug effect between the solid symbols here and the open symbols. And we can see the time course of this effect comes on relatively quickly and is relatively constant after just a few days. So from this, the traditional analysis that's done in these phase 3 programs is to look at the last week of the pain of the clinical trial and looking at the difference between placebo and active doses. And it ignores all the other information along the way. Conversely, if we look at this in a longitudinal fashion, we can then look at take into account all kinds of other types of information, biological information that, for example, would be important, like such as the dose response across different doses in the trial. This drug, gabapentin, has dose-dependent bioavailability, so it's important to take this into account. And really, you're using then all the information over time, as opposed to ignoring, let's say, 90+% of the information that's gathered within the during the study. This is an interesting story. Part of the reason why we included it in that particular publication is that it had a major impact on regulatory decision making. So the model was used by regulators. We had 2 pivotal trials, but they used different doses, and they were going to request us to perform a 3rd trial to replicate the efficacy at the doses that were used in the previous 2 studies. So the model was used to confirm that the 2 pivotal trials would produce a similar response if the doses had been the same. Then the US FDA included the following statement in the clinical study section of the label, and this was quite precedent setting at the time, that pharmacokinetic, pharmacodynamic modeling provided confirmatory evidence of efficacy across all doses. And we avoided the need to perform another clinical trial in the support of the approval and label for gabapentin in this type of neuropathic pain and post-herpetic neuralgia. So a very impactful example going back, I guess some 16, 17 years ago. I touched on this briefly earlier, that the term has evolved over time. When we, so we'll see terms in this presentation about pharmacometric systems pharmacology. Many of you are very familiar with these terms. When we wrote this paper in 2007, the term model-based drug development was used. It was used in a document that came from FDA in 2004. So this has evolved over the years to model-informed drug development, model-informed drug development and drug discovery and development. So sometimes MIDD, MID3. Essentially, I'll be using these terms more or less interchangeably here. And this is just to show you some of the history behind some of these this terminology. So back to this publication and some of the other examples. I'll focus about the components about meta-analysis here. So this is an example of a meta-analysis, a network meta-analysis, where we want to look now at the comparative efficacy from clinical trials in a particular therapeutic area in the setting of venous thromboembolism prevention in people with different types of orthopedic surgeries. But this is the point of this slide here, is that we can look at direct comparison. Let's say apixaban versus enoxaparin, or rivaroxaban versus enoxaparin. So the number connecting these symbols here is the number of clinical trials that are available in this analysis. But when we do a network meta-analysis, or a certain specific type of network meta-analysis that we refer to as model-based meta-analysis, we can, because we're quantitatively essentially extracting information on both the direct comparison, but also the indirect comparison of, let's say, apixaban and rivaroxaban, or warfarin and dabigatran, even though there may not be any direct comparison because they have a common, say we have a common comparator. So we can extract quantitatively information on the comparative efficacy by an appropriate type of meta-analysis. And this is very useful in how we make judgments in drug development based on a quantitative information, again, of the prior data, comparative efficacy. So from this, we can generate figures like the one showed here, where we can look now at the probability of outcome of a proposed trial. So we could be designing a future study, and with different sample sizes, this would be a very large study. We could look at the probability, for example, of being superior if we have an active comparator in the study, or the being non-inferior, probably of being inconclusive if we have too much uncertainty, or the probability of being inferior. And you can see that as you increase the sample size, you may have gain greater confidence in certain, the likelihood that you'll be able to show, let's say, superiority or non-inferiority. And you can see how essentially, if you double the size of the study, which may have major cost implications, how much is that getting you in terms of value, in terms of confidence in what you'll be able to demonstrate. So we use this information along with some quantitative criteria. Traditionally in drug development, we've very often focused on, is the drug active? Are we beating placebo at a p-value less than 0.01 or 0.05? And what we've wanted to change here is the mindset towards differentiating and really looking at the probability of technical success, if you wish, shown here, where these bars may, so for example, reflect the probability distribution, if you wish, of a particular drug response relative to a target value, maybe what we need that's important, and lower reference value may be, for example, a placebo or an active comparator. But if we have great deal of confidence that we are above the target value, we would, for example, go forward. You may pause if you have a lot of uncertainty. You may have a drug that's effective, but you don't have enough precision, so you may gather more information. And these are examples here where you may want to stop, where you have insufficient evidence that you can achieve the target value that you need for clinical and regulatory success, and especially for reimbursement decisions. And this is an example here where we may have a drug, for example, that is effective. So it may be better, let's say, than this is placebo, this vertical line here. We have an effective drug, but it's insufficiently effective to be for clinical and reimbursement success. And that's really changing the mindset in terms of how we've approached decision-making based on these types of probabilities, as opposed to, are we beating placebo with a certain p-value? Focusing a little bit more on this topic for a couple of minutes before I get into examples. Matt Zayert here was in our organization at Pfizer, and along with several other colleagues, wrote a paper on some of these concepts about statistical power versus the probability of trial success, that we should not ignore the probability of success and use only statistical power. The traditional power assumed a fixed effect size, as if we that we have no uncertainty. So we assume an effect size, we assume our drug is effective to a certain level, and we can design a study accordingly. Conversely, the probability that a trial will be successful tries to account for this expected treatment effect and the uncertainty. So we can think of this as really an unconditional probability. So an example here, going back to the representativeness bias that I mentioned earlier, is that we may have a prior signal, maybe from a proof of concept study, where we may have a certain amount of uncertainty, and people will often very much focus on the mean response and ignore that uncertainty. So we may have a situation, if there's a lot of uncertainty, that the probability of success of POS could be very low, despite having a study with very high statistical power. And this could be part of the reason why we have some low success rates in phases 2 and 3 clinical development, that people may be getting anchored with the statistical power. In other words, they think that we have they know we have a study that's powered to 80 or 90%. They should know better that that's not the probability the trial will be positive, but they get anchored from this and they don't adjust sufficiently as they should for the uncertainty. As this leads to a common discussion that we would have on some of our governance committees, when somebody would say, well, I'm surprised that this last trial that we conducted was negative. And the appropriate answer would be, well, I'm surprised that you're surprised, because we only had maybe a POS of 61%. So it was almost a coin flip that the trial was going to be positive. So graphically, this is shown here, where we can look at this probability distribution. Now, this is based on the prior data. You could be building one of these meta-analyses, if you wish, or a model, to say that we have the probability, in this case, some 84% in the shaded blue area, that our response, the effect is better than 0. So we have a positive effect. This is just about the prior data. This is the state of our knowledge, let's say, at a particular point in time. Now we're designing a future study, and we may design a study and say, well, at this most likely effect, we may have a study with a certain sample size, and say, well, we'll have a power of 90% at an expected effect of 0.5. So this is assuming the effect is exactly 0.5, but we have some uncertainty in this effect here. So the power, if the effect turns out to be 1 or 2, will be higher, shown by the red line here, and the power would be actually lower if the effect turns out to be less than 0.5 or 0, or even less than 0. So the power would be less. So the way of looking at this is to look at this, if you wish, integrating this statistical power over all these potential values of the treatment effect. And that gets you to what's shown here. At the bottom here, where the probability of success is some 66%, even though the study was powered to 90%. So we've, over time, basically required that our major clinical trials always come with a probability of success estimate. So people were making these decisions with eyes wide open. It's fine to take a risk, but you want to make sure that people know the risk that they're taking when a large clinical trial is being proposed. And if you know, some of these clinical trials could be costing millions, tens of millions, and hundreds of millions of dollars for large phase 3 trials, even in some cases over $1 billion. So the key point of this is that statistical power of a trial is not the same thing as the probability of success of a trial, and you want to guard against being anchored too much on the statistical power, and really always also report the probability of success of a trial when you're trying to plan a future study. So you can use this information here to look at, for example, the probability of success of a proposed phase 3 trial. Let's say if our criterion is, let's say we just want to show efficacy relative to placebo, and we may have a dose response model to see that across different doses, we'll have different probabilities of success. And as we change the sample size, so it gives you an idea how much benefit you have, if you wish, in terms of increasing the probability of success, how much value you get for that increased cost of the study. So a way to quantitatively look at this information. So in 2013, our organization looked at the impact of MIDD. So there's a publication by many of our colleagues at the time led by Peter Milligan, looking at the impact across many different programs. We called it still model-based drug development then, and the efficiencies that were gained across many therapeutic areas. So this was to show again the impact on key decisions. And for the sake of time, I'll just focus on one here. An example of the design of a phase 2B dose ranging study in the setting of venous thromboembolism prophylaxis in patients undergoing elective knee replacement. So patients who get the knee replacement are at risk of blood clots. We typically give them prophylactic anticoagulants, and this is a very challenging area because we have to balance the risk of underdosing, because that would give an increased risk of thrombosis or blood clots. And if we give too high of a dose, there will be a risk of bleeding. But at the same time, we want to study a wide range of doses because we want to estimate doses for future drug development. So the objective of this program at this stage was to find a dose equivalent to the current standard of care, which is a low molecular weight heparin, enoxaparin, 60 milligrams once a day. And this was investigating an oral factor Xa inhibitor. So these were some of the prior data that we had with some biomarkers and data from competitors. We could look at the probability of VTE. Again, these are the blood clots, if you wish, across the different doses. So the probability goes down, so the efficacy essentially goes up. You're having a lower risk of blood clots. That's what you want as you increase the dose. And on the flip side, as you increase the dose here, you get an increased risk of bleeding. As you can see, there's a lot of uncertainty in these relationships here. So we want to take that into account, but we can use this type of model to then conduct clinical trial simulations where we use this VTE and the major bleed dose response models to simulate numerous clinical trials to evaluate design. And we are trying to focus on some key questions, which is the power to find a dose equivalent to enoxaparin, the standard of care. But also we wanted to conduct most likely an adaptive trial at this stage, and we knew we would have to add or eliminate doses based on the information that we acquired during the study, because we also want to protect subjects from excessive blood clots and excessive bleeding while evaluating the dose-response relationship. So it's a dual challenge, if you wish. And we also evaluate the sensitivity to all kinds of factors in our study design, the sample size, the doses, how to adjust to add or prune or delete doses. And as I said, the goal was to select one dose for the phase 3 clinical trial. So we focused eventually on an adaptive dose ranging study. It was a 6-arm parallel group study. It was an adaptive study where we started with 5 doses of the investigational agent from 0.1 to 2.5 milligrams once a day. We would eliminate doses based on excessive VTE or major bleed. Eventually, we added higher doses progressively as the trial went on, as we gathered information about the dose response and the risk of major bleed and VTE. We saw that at these doses, the major bleeding event was within our predefined criteria. And enoxaparin, as I said, was the active control. We evaluated through our simulation that we needed about 147 patients to have sufficient confidence to make a decision about adding or deleting doses. And our total sample size was 1,250 patients. I'm summarizing here probably a couple years worth of work in about 3 minutes. So the impact in this case here of this adaptive trial design is the study was designed using modeling simulation. It was approved by senior management and was conducted successfully. We met the key objective, identified the dose of that was equivalent to enoxaparin with good precision. We safely explored a 100-fold dose range. So we were able to really characterize the dose-response relationship quite well. As I mentioned earlier, this is quite challenging, and we see in the among publications, often people are quite restricted in the dose range they study because they're quite concerned about it going too high or too low. So it was a successful trial. It was also successful in the sense that we used a third of the sample size of the traditional parallel group study in terms of number of patients, cost, and especially time to conduct the trial. So a very good example that was summarized in that publication. I'd like to take now, switch to a much more recent example. This is one that actually is available online to anyone here with an internet connection. This is a journal that is freely available, as many of you know. And this is about the end-to-end application of model-informed drug development for ertugliflozin, a novel sodium glucose cotransporter 2 inhibitor, or SGLT2 inhibitor. This is a program that was done at Pfizer. These are colleagues from our global clinical pharmacology organization, started for many, many years when I was there. This publication was just accepted just a few weeks ago, but it's available online. So it summarizes nicely the work that was done. So an SGLT2 inhibitor works by blocking the reuptake of glucose in the kidney. So it leads to glucose excretion, more than you normally would have. Normally would have little or no glucose excretion. So that's a great biomarker. So this is the UGE, this is urinary glucose excretion over 24 hours. In the first in human study, so we're using it as a biomarker of the pharmacology here. So this is the dose escalation, typical in the first in human study. And what's shown in the dotted line in red here are the model predictions. So this was done using a systems pharmacology model that was developed based on a lot of prior information in this particular therapeutic area, looking at expected response, and we picked our doses accordingly. And in the shaded bars here are the observed results that we saw in our first in human study with this biomarker. Again, very consistent with our model predictions. A precedented area, so that always worked nicely. We also had this information to then predict what we would see in phase 2. Now, this is a very different endpoint. This is now from healthy subjects to patients with type 2 diabetes. So we're linking this information. Recall this continuum that I mentioned in the slide earlier from, let's say, preclinical development to registration. I'm going to walk you through the continuum here on a project that took several years. So now we have a different endpoint. We're looking at the placebo-adjusted change in glycosylated hemoglobin. So this is now a phase 2 study in patients. And this is a study, a 12-week study, looking at doses of ertugliflozin, the model prediction, showing the data in the shaded area or the colored area, with the observed results from the phase 2 study. So the point of this slide here is that we could develop this model to then predict what we would see in a completely different endpoint, different time frame, different subject population from phase 1 to phase 2. As we went forward, we used this modeling to then select doses that we want to take going forward. So we selected the doses of 5 and 15 milligrams. These were the responses in terms of glycosylated hemoglobin. So these are clinically significant changes in response in patients with diabetes. So these were the data now from the phase 2 study. Eventually, we conducted a study in phase 3 at this 5 and 15 milligram dose. And now integrating in the phase 2 and phase 3 data, we see a similar response here in terms of over a longer time frame now, the response in terms of glycosylated hemoglobin. This is the registrational endpoint. We could further take in the drug development program now, the population pharmacokinetic information that we have. So now we're integrating the data from phase 1, phase 2, and phase 3 to look at the impact of different patient factors, covariates, if you wish, like renal function, the presence of type 2 diabetes, gender or sex, and different race groups, to see what is the impact on drug exposure, which is reported here as a ratio of AUC relative to the reference. So 1 is when there's no change. So this is showing an increase in exposure or a decrease in exposure. So a way to make judgments about the impact of different covariates. So this is integrating all the information again on population PK, and also relating this to what we know is the therapeutic index of the drug based on our clinical studies to show that these differences are important or not very important, given the safety and efficacy of the compound across a broad range of doses. So this is the population PK across phases 1, 2, and 3. Finally, as we moved into late stage development, there were questions about drug-drug interactions that needed to be evaluated. This drug is eliminated via glucuronidation. There's a group of enzymes we call the UGTs. They're responsible for the clearance of reticuloflosin. And there are some known inhibitors of these enzymes, one of them being mefenic acid. So again, what we did here was that we used physiologically based pharmacokinetic modeling to develop a simulation, if you wish, prediction of what would be the expected impact of giving reticuloflosin with an inhibitor of UGTs. Again, we had some prior information to base this on, the in vitro metabolism, the PK of the drug, and we could estimate what would be the AUC ratio, for example, in the presence of mefenic acid. So the key part is what I highlighted in yellow here, is that the HER2 PBPK results were submitted to the US Food and Drug Administration, and a clinical UGT DDI study was not needed, and the results were incorporated in the DDI section of the label. So this is an example of where we use modeling simulation instead of doing a clinical pharmacology study. So I'm getting close to wrapping up here, just a few more minutes. In 2005, I gave a presentation at the American Society for Clinical Pharmacology and Therapeutics meeting, and on some of these principles. And my concluding slide, I said, imagine where we can be in the future with what we said at that time, modeling and simulation, now MIDD, as we have libraries and models for all our key therapeutic areas. Our trials and development plans are designed using modeling and simulation. And we can extend this not just in clinical development to the preclinical phase, leveraging more preclinical and discovery information, and also in the post-approval health economics by linking pharmacometrics and pharmacoeconomic models. So a lot of work has happened since then, and I'm just going to just show a few for the sake of time, cite a few papers to highlight how this has progressed over these 16 plus years, actually, or longer. So this is a publication from FDA looking at the impact on modeling pharmacometric analyses on approval and labeling decisions. This is during the 2000-2008 period. This is a more recent publication from 2019 looking at physiologically based pharmacokinetic modeling and the tremendous growth in the application of PBPK in drug development and regulatory decision-making. This is a very nice best practice document written by Scott Marshall and a collection of from our global clinical pharmacology organization at Pfizer, but also really a group of European colleagues based mostly in industry and describing MIDD practice, applications, and documentation. So a great paper if you want to read more about this. This is a current US practice and future consideration paper with from the leaders in the clinical pharmacology organization at Pfizer and the pharmacometrics organization, at FDA. Another example here of model-informed drug discovery and development, looking at current industry practice and regulatory expectations. So this is a publication again by Scott Marshall at Pfizer, but with colleagues both from US FDA and the EMA, looking again at regulatory expectations. There is a model-informed drug development pilot program at the FDA where industry people can take advantage of interacting with the FDA about their MIDD strategy. I'll come back to this in a second. And finally, this is an example here that speaks to my last bullet in 2005 about linking pharmacometric and pharmacoeconomic models. This is a publication that's in press now in pharmacometrics and systems pharmacology, linking these different disciplines, if you wish, by colleagues in academia and colleagues at Pfizer. So very nice, that's a publication that's in press. And very hot off the press, just to give you another very recent example of MIDD in drug development. So this was April 23rd, so about a month ago. There was a statement, FDA approved a new dose of this drug for the treatment of tardive dyskinesia based on model-informed drug development. So this was an example here where a new dose was approved without the need to conduct clinical trials based on MIDD principles. So again, a very recent example. So in conclusion, drug development is a sequential process over 10 plus years from discovery to registration. Most drugs fail in drug development. We have common biases in drug development, in making decisions under uncertainty. This happens in all kinds of different decision making, certainly is applicable in drug development. And we can view drug development as a model building process that takes time, where we continuously build this information. So MIDD is useful to incorporate new information gathered over time, that continuum that I talked about. We integrate data from different sources into a quantitative framework. Our models can be in very different varieties, be based on PK/PD principles, systems pharmacology, pharmacometrics, could be meta-analysis of clinical trials, combining some of these different elements. The key point is that we want to leverage the totality of information instead of selecting particular source of data that fit, let's say, our preconceived ideas. Again, dealing with this confirmation bias that I mentioned earlier. These modeling approaches really allow us to be more transparent about the assumptions and uncertainties. That should be a strength of these approaches. Can inform drug development strategy and trial design. I think I gave you some examples of this. And I gave you also examples where we can potentially avoid studies via simulations. And finally, that MIDD has evolved and progressed over the past 15 years through many different examples. I only cited a few here, but there are literally dozens and dozens of examples of impactful stories on both drug development strategy, trial design, and regulatory decision-making. So with that, I thank you again for your attention, especially those of you who are listening to this late in the evening. And I think we may have time to entertain some questions. So again, thank you very much.
Thank you, Rick. That was an awesome presentation. And I can see that this presentation really encompassed your breadth and depth of expertise, and covered so many aspects. And I myself was looking at the number of review papers and the articles and references that you have shared. Just amazing, amazing. And I wanted to, we will start taking questions in a couple minutes, and I wanted to share a few things with all the participants, that today's presentation is being recorded, and after, I would say, a day, we would actually have this presentation on Project Dontabhaktuni's YouTube channel. So you will have the option to go back and revisit today's talk. We will also share with you the slide deck of today's presentation. So you will have the opportunity to go back, look up the references and read them at your leisure. So expanding your knowledge and learning more about model-informed drug development. Without further ado, I would call upon Arun to start taking on the questions. We have a few questions in the chat right now.
Thank you, Dr. Lano. That was a wonderful session. We have a few questions from the participants. So the first question is, how much effective it would be to develop a model of neuronal diseases, as it is quite difficult to understand the complete pathologies and replicate them in model for neurodegenerative diseases?
So you're right. Neuroscience and neurodegenerative diseases are particularly challenging. And that's evidenced by the common failures that we've seen in drug development, for example, in Alzheimer's disease over several decades, really not finding drugs that are effective at reducing the progression of the disease. My answer to your question about will it be useful, I mean, I think it's always useful to develop a model. It allows you to integrate the information that you have. It allows you to know what you know and also what you don't know. So it can help you, for example, determine, well, maybe there's a gap in our understanding here, and this is how we should try to develop further evidence to how to fill this gap. There should be no illusion, though, that models don't create in of themselves. We won't find a drug that's effective for Alzheimer's disease just by reanalyzing data. It just allows us to get some insights. From these insights, we can, let's say, a mechanistic model. If we look at, really a systems pharmacology model, we can sometimes find out, for example, where in a very complex cascade, where if we want to perturb a particular cascade, what is the expected effect might be. And we've gotten some great examples of this in systems pharmacology, where we found out, for example, that even if we were to have a drug that is very effective at particular, let's say, blocking a certain enzyme, it will not lead to the therapeutic effect that we want in a particular disease of interest. So there's always some useful information to glean from developing these models. I want to caution anybody, like I said, my point of this presentation here is not that, as the old joke would say, is that, well, if we model the data enough, we can turn an ineffective drug into an effective one. That's not the point. The point is to how to integrate this information as best we can, not ignoring parts of the information. When we develop models, we typically look at data from many different sources, especially in these systems pharmacology models. There's always something to be gained in terms of the knowledge and insights. How ultimately useful it will be, it's very difficult to predict. And I think in the case of neurodegenerative diseases, I think there's no doubt that this is a very, very, very risky part of drug development, but there's always some information to be gleaned from this. So I would encourage people, if you're working on this, if you share it, I think that's, it can always add to the body of knowledge, but caution you that it's not necessarily the, in of itself, let's say the key to unlocking a new therapy for Alzheimer's disease, for example. But it can certainly be helpful in gaining in our understanding, and incrementally over time, we will get to crack these more difficult nuts of these more challenging diseases. It's just the process may be longer than other diseases where we may have a lot more examples, let's say, where the biology is a little bit more, is less complex, shall we say.
Thank you, Rick. Arun, we do have more questions.
Yeah.
So.
Yeah. Moving on to the next question. So we have a question from Vaibhav. So with a regulatory standpoint, at this point, we have modeling and simulation acceptance, which is restricted only to the interpolation of dose. So what is your take on how it would be changing over the years, and the extrapolated dose regimen will be accepted based on modeling and simulation activities only? Will it be accepted only based on modeling and simulation?
That's a very good question, actually. So you're right. A lot of the examples are, some of these are interpolation. The very last example I showed you from tardive dyskinesia was an interpolation. So it's obviously more challenging when you get into the extrapolation. I would say that some of the, if you're talking about pivotal safety efficacy study, that's a much taller order, I would say, to get to, because one of the challenges with models is that we can model what we know. For example, let's say the efficacy of a drug. There may also always be some unknown unknowns, if you wish, something that you have nothing that's incorporated in your model that you may observe. And that's why the empirical approach, I think, of demonstrating this through a clinical trial has so much power. But there are definitely some examples of where we've avoided clinical trials completely. I gave you the example of gabapentin earlier. So that was a cross-validation across different doses, if you wish. The PBPK examples are where we are completely avoiding studies. It's essentially it's an extrapolation, if you wish, in the sense that we are not doing a clinical study, and what's put in the label is a simulated response, not an interpolation, really. But I take your word that, and I agree with you that for all of the examples that we have, there'll be more in the domain like I just described, as opposed to, let's say, a phase 3 program where we did say, extrapolate, especially to higher doses via simulation. That's a taller order, partly for the reason I mentioned in terms of the unknown unknown. I think it's useful in science, and for those of us working in this discipline, to keep this in mind, that our models, while very useful, and obviously that's what I've been talking about, and it's been my passion for decades, we always have to keep in mind that there's some parts, the model only help us for what we put into it. There could be some unknown there that is not captured in our model, that with time, if we do more studies, we'll be able to capture it, but it may be unknown at a certain point in time. And as we conduct extrapolations, that would encourage caution. So as one of my statistician colleagues would often remind us, and I think it's useful, when we look at a model prediction, you have to keep in mind always is that it's under the assumption that the model is correct. If we have a model that has not been properly validated, if you wish, or the model predictions may look very good, and we've seen examples of this in my experience, where we have a nice model prediction and we observe results that are outside of those predictions, completely outside the range of uncertainty. And basically it tells us that there were some uncertainties, some unknowns that we did not incorporate in our model. Now, the lesson from this is that now we add this new information, now we update our model to better understand this uncertainty. But I think it's always useful to have a bit of humility in how we go about making these predictions, especially when we get into extrapolation. So point well taken on the extrapolation beyond, let's say, doses that have been evaluated.
Thanks, Dr. Leno. There is another question, which is the data from model-informed drug development acceptable by all the regulatory agencies?
That's also a good question. So I gave you a few publications there that focused on the European regulatory, the EMA. the US FDA. I've been involved in programs like this in Japan, in China. But it would be, so as you probably all know, certain regulatory agencies are going to be, more influential even beyond their borders. So I think when Europeans and American regulatory agencies accept a certain approach that carries a certain amount of weight with other regulatory agencies, but it would also be fair to say that it's not certainly a one-to-one relationship. So we've always seen gaps, for example, in Japan and China, and actually even within a regulatory agency. Within the US FDA, for example, those of you who work with the FDA, we often comment on how when we deal with different medical divisions, there may be relatively more conservatism or more willingness to accept different approaches. So I think the short answer to your question is that this type of information, there's enough precedents now, I think, that have been used across all the, let's say, essentially all the major regulatory agencies, but it can be variable to some extent, and it can be variable even within a regulatory agency, like in the US. So I would say part of why I chose the examples that I chose is I want to make sure that as these examples of impact, that we put them in the public domain, because we want these examples to be then used to support further, to go beyond, let's say, where the particular regulatory decision that was made. So to use them, and as we publish more of these examples, and there's a lot of sharing between regulatory agencies, I think this will broaden a more consistent application. It takes time. It's an evolution as opposed to a revolution. But I would say we are in a very, very different place today than we were 10 years ago, let alone 20 years ago. At FDA, for example, with the MIDD pilot program, as I shown in that, I think the second to last slide, the FDA is actually showing examples of where they used MIDD to make their decisions. So we don't even have to promote this; they're doing this themselves, and I think this will continue to have more influence across the world. But I've received very receptive audiences. I remember spending an extended period of time at the Chinese Food and Drug Administration some 10 plus years ago, describing some of these principles. So I think there was a willingness to move in this direction, and that it will be a continuous evolution.
Thank you, Dr. Lalonde. The next question is, how much modeling and simulation is relevant or helpful when the drug moves from preclinical towards human trials?
So I understand the question, how much is modeling and simulation useful when we move from preclinical to clinical? I mean, it can be tremendously helpful. The example I gave you on ertugliflozin allowed us to move very quickly through the phase 1 program, because we could leverage a lot of the preclinical PK/PD information that we had, and also the competitor data that we had, to select doses, make sure we had a reasonable biomarker, and then how we could quickly progress when the biomarker response was essentially as we expected, and the toleration was as we expected, or I'd hope maybe. So the translation from preclinical species to humans, there's a tremendous opportunity there. I focus on the PK/PD. There's the simple PK part also, making sure we have ways of predicting exposure and doses in humans. But on the PK/PD side, can be tremendously useful for selecting doses and accelerating programs, let's say, where we feel we have a little bit more confidence. I should quickly add here a common area where we have a lot of confidence would be is a precedented mechanism. So if we have other compounds that have been tested in humans previously, and we're bringing a compound from a similar pharmacology, So versus a completely unprecedented one where there would be a little bit more uncertainty, but then again, where we can maybe, if we have a good preclinical model. At the first in human, as you probably all know, we have a lot of uncertainty. So we typically study a very wide range of doses, start at a very safe dose. So even if there's some uncertainty about some of these relationships, the gain in knowledge is exponentially greater as we just start administering the drug to the first few cohorts of a first in human study, because we have much more convincing data on the pharmacokinetics and hopefully on the biomarker response, confirming what we may or may not have seen, let's say, in the preclinical species or some other in vitro model, or from a systems pharmacology model that we may have developed. So long way to say that I think in the slides that I showed about the continuum, I think it's very every one of those stages. In that last publication on reticular flow, and the reason I included it, it talks very much about that beginning to end application of MIDD from the preclinical stage to design the first in human study, all the way to the impact on labeling with the drug-drug interaction data and everything in between.
Thank you, Dr. Leno, for answering the questions. That's all we have from the chat panel for the questions.
Thank you, Rick. Thank you so much for being patiently answering all the questions. And as I had mentioned at the beginning of the talk, that I actually would also like to present some more details of our upcoming workshops. And Rick, if you stop sharing, I'm going to start sharing my screen, and I can share with our audience today to tell them about our coming workshops. So next month, we actually would be hosting a 3-month program, which is going to be a certified program in pharmacometrics and PK/PD modeling. And in this workshop, we also will be touching upon the RS-NLME, which is R Speaks NLME package, a new tool from Certara. In the next few days, we would be making announcements on how one can apply for this program. There would be a prerequisite that you will have to fulfill on in order to become eligible to participate in this program. This program is going to be focused mainly for participants from Asia. There may be some exceptions for participants from United States. However, we are focusing this more for participants from Asia who have few opportunities to expand or learn pharmacometrics. Again, a workshop of this size and magnitude is not possible without the contribution from many, many people. Again, I really thank Rick for making the time and presenting this morning. And our board members, Somdeep Datta, Parthanandi, and Rajan, who have been great advocates, who have supported Project Dontabhaktuni through the various patches that we are going through. We are still a small organization trying to make a contribution and make a difference in clinical pharmacology and pharmacometrics knowledge sharing. And of course, the multiple volunteers who have been playing a critical role in the background, reaching out to you, sending you emails, and encouraging participation. Ramya, Krishna, Ramesh, Arun, to name a few. Again, Also want to acknowledge our sponsor. So today's workshop is sponsored by PharmaPro Consulting. PharmaPro Consulting is a unique consulting company that currently has over 25 experts in the group. Majority of the team has over 20 years of experience, and they support data management, PK/PD modeling, strategic advice, pharmacometric and PAP-PK modeling, study design, regulatory affairs, and CMC. In the past 6 years, the company has worked with many big and small pharmaceutical companies. This company is certified as a women-owned and a minority women-owned organization in New Jersey, as well as by the federal government. And again, leaving you with the thought, do consider participating in our 3-month hands-on certified program in pharmacometrics and PK/PD. And please stand by to hear more about the registration process for this workshop. Again, this is going to be driven based on certain aspects, and there is a registration process that one will have to apply and get approved before you can participate in the program. And today's workshop, as well as our past workshops, you can find them on our YouTube channel. You will have access to the slide decks which have been presented and the references as well at our YouTube channel. Thank you for being here, and thank you for giving us this opportunity to share another of these workshops with you.